Okay, so we've had our solutions sitting here for the last 15 to 20 minutes, and there's a couple of things that I'm going to point out that aren't going to be as obvious to you because of the quality of the video that we're dealing with, but in general, if you look at the DI water, of course, you can see, I think you can see the type through that. At 100 milliosmoles in that solution, blood also begins to clear, but it still remains somewhat opaque. I can see the letters, but I can't read the words through the 100 milliosmol solution. At 150, I can't really see the letters very well unless I slide the tray back and forth a little, and then I can see some motion in the bottom as the light bounces off from the paper. I can see that there's something there, but I don't have any idea what it is. Now, by the time we get to 200 and 250, 300, 350, we don't see any changes after that. Okay, and so what's happened then is that the we've discovered the isoosmotic point is being somewhere between 150 and 200. Now, if we were to measure that in human blood on an osmometer, we would have an osmolality of roughly 180 to maybe 220, somewhere in there. And so our experiment then gives us some gross measures of what the isoosmotic point is. And that's an important point to know. Obviously, if you're trying to inject someone with medication, for example, and you don't have the right osmotic concentration, you can cause their red blood cells to burst. And so there are problems associated with that. Mostly we've learned all of those things the hard way early on before we understood things like isoosmotic and isotonic, and that those are two different, they sound similar in how they work, but they have two very different results. And that's what we're looking at here. So when we have a situation where you have an isoosmotic solution, in nearly all cases, it's also isotonic. Keep in mind, and you've all read the handout that I've sent by now. And so isotonic just refers to how the cell responds. Does it crenate? Does it remain the same? Or does it hemolyze? Does it burst? Okay, and so those are the things that happen when we talk about isotonicity, right? So how the cell responds. Isoosmotic means that you have the same number of osmotically active particles on either side of the membrane, so there shouldn't be any net movement of water from one compartment to another. It is, however, I'm going to go ahead and give you this little tidbit of information, which sounds completely counterintuitive to everything that we've learned so far. It is possible to create an isoosmotic solution that is not isotonic. And you might be asking yourself, well, how can that possibly be, right? If you have the same number of osmotically active particles on either side of the cell membrane, there should be no net movement of water from one side to the other. And that is true for 99.9% of the things that are out there. But in cases where your osmotically active particles are able to cross the cell membrane, because its concentration gradient is so high outside compared to inside, you can see the cells burst in an isoosmotic solution. Now, the one that we usually do in lab is with urea. I make an isotonic solution of urea. We put the cells in that isotonic solution, and just like DI water, they burst almost immediately. And the reason that happens is not for the same reason that it happened with DI water, which just finds its way in and follows the concentration gradient of salt, but rather the fact that urea never occurs in cells, but the cell membranes are extremely permeable to urea. So in this particular case, when we have an isoosmotic solution of urea, and we put red blood cells into that solution, the fact that the driving pressure, the gradient basically, the driving gradient, forces urea into the cell, that means that you've added some isoosmotically active or osmotically active particles to the interior of the cell. Water follows. And so again, this has some applications for certain types of medications, right, where you might inject them in various ways. So we have to always be cognizant of the fact that even though it's isoosmotic, doesn't mean that it's isotonic. And so it seems counterintuitive, but think about that, and I think you'll get the idea. Now, ordinarily, what we would have done in the lab for the next hour and a half was we would have sat there with a microscope, and we can use an ocular scale and in the ocular lens to measure, they call it the ocular micrometer. We can measure the diameter of cells. And if you were able to see them, and I put into the handout that you should have received, I put in a drawing there that I shamelessly swiped from the internet that shows hemolysis and crenation. And so you can see that in those situations where the cells taking on water, they get quite round. Ordinarily, red blood cells are relatively flat, got a little they look like little donuts almost. They've got a little compressed area in the middle, for example. And that's the usual state that you see them. But if you put them in a solution where they pick up water, they get very globose, okay? And if the solution is strong enough, they will burst. And of course, then DI water does that. Ordinarily, if you were here in the lab, I would have asked you to put a drop of blood into the DI water and then put it on a slide. And then I'd let you sit there for a few minutes until somebody came up with we can't see any red blood cells. And yeah, you can't, because they burst nearly immediately. And all you see is just the red clear fluid. So we have successfully determined pretty close, really. And of course, we could have made up many, many more solutions and refined our precision on that measure. But we were able to go ahead and make a rough guess at what the isoosmotic condition is then for red blood cells. And that was really the goal. And of course, being aware that this process is not applicable to things that have high cell membrane permeability, but do not occur in high concentrations in the plasma. And so in those cases, you end up with that urea example that we talked about, where even though it's isoosmotic, the isotonicity on that is, well, it's not isotonic. It's in fact going to act just like DI water and cause all the cells to burst. So that's an important component. What I need for you to do then is to put together a brief lab, I guess I would call it a review of what it was that we did, what the objectives were. I think that all of that's listed on the back pages of the handout that you've got. Please fill that out, and when you get that filled out, and if you have any questions about the experiment that we've done here today, certainly you can write to myself or to Tyler, and we will be more than happy to explain that. But do take some time and study this particular process. We won't really talk about it too much anymore, although in labs that in the lecture materials that I'm handing out, or that I'm putting up for you to look at, there will be opportunities to see some of the processes that we've talked about here in lab, in some of the ways that may affect how your body reacts, and in this case, cell membranes work, right? And that's what we're really after. Cell membranes are really complex things, and we're trying to get through this part so that we can begin to see why it is that cell membranes are so versatile that we can have specialized cell membranes like what we might see on neurons, and then we just have fairly simple cell membranes like we see in red blood cells, which basically are designed to just hold in hemoglobin and carry oxygen, and that's pretty much their only function. So again, make sure that you've completed all of the components that are in the handout that's out. You can take pictures of those, or you can put them, scan them, whatever works Tyler. And I think that will conclude our first attempt. I'm going to say it's not a great one, but it's the best attempt we could make with the conditions that we're under. So everybody be careful, and we'll put together another lab for next week.